AS Biology. Welcome. Are you excited for a fun learning session? Make sure you have your book and a pen before we start. Today you are going to learn about RNA and DNA. DNA and RNA. DNA, deoxyribonucleic acid, is a double-stranded molecule. The DNA molecule is said to have a double helix shape as each strand is a helix. The DNA strands are held together by pairs of bases. Between the bases are hydrogen bonds. The backbone of the DNA molecule is made up of an alternating sugar group and a phosphate group. Nucleic acids are found in all living things. Both DNA and RNA are type of nucleic acid that have different functions. DNA is used to store genetic information. RNA is used to transfer this information from the DNA to the ribosomes in the cytoplasm. Ribosomes are the sites of protein synthesis in the cell. A nucleotide is a monomer, and when they are bonded together they form DNA and RNA. Nucleotides are made from a pentose sugar, a sugar with 5 carbons, a nitrogenous base, a phosphate group. The structure of a nucleotide is shown. In DNA the base can be adenine, thymine, cytosine, guanine, and the sugar is a deoxyribose sugar. In RNA the base can be adenine, uracil, cytosine, guanine, and the sugar is a ribose sugar. The ester bond is formed between the pentose sugar of one nucleotide and the phosphate group of the next nucleotide. The reaction that bonds the nucleotides together is a condensation reaction, which means that a water molecule is formed. If the DNA strands are both the same way up, the bases do not touch. Therefore the strands run antiparallel to ensure that the bases are in the middle together. This means that one of the strands is upside down. Bases held together by hydrogen bonds. This holds the strands together. Antiparallel means that the strands are running in opposite directions to each other. One is upside down. These strands twist around each other to give the double helix shape, the shape that was discovered by Watson and Crick from a photo taken by Rosalind Franklin. RNA is single-stranded, shorter than DNA. Ribose sugar in the nucleotide bases A, UC, G. DNA is double-stranded, longer than RNA. Deoxyribose sugar in the nucleotide bases A, TC, G. There are 4 bases in DNA, and these can only bond in a specific way. We say that the bases are complementary. The 4 bases pairs are adenine, thymine, cytosine, guanine. The bases are complementary, pair in a certain way, and they will only bond as follows: adenine to thymine, cytosine to guanine. One way to remember this is apples grow on trees, cars are stored in garages. The reason that base pairs are complementary is because there are a specific number of hydrogen bonds that form between the 2 sets of bases. Between A and T there are 2 hydrogen bonds. C and G there are 3 hydrogen bonds. Because there are equal numbers of A, T, this means that if you know the percentage of A, you know the percentage of T. This is also the same for C, G. For example, if a sample of DNA is 32% adenine, work out the percentages of thymine, guanine, and cytosine. For example, if a sample of DNA is 32% adenine, work out the percentages of thymine, guanine, and cytosine. If A equals 32, the T must be equal to 32%. To find G and C, add these together. 32 plus 32 equals 64. Take away from 100% equals 100 minus 64 equals 36. 36% is the total amount of C and G, so each must be half of this sample, 18% G and 18% C.